All right, thanks very much, Paul, and thanks very much to the organizers of the challenge for the privilege to be part of this community and the privilege to be the one to curate and sort of select the data to torture the participants with. I'm very passionate about this field of predicting AMR phenotypes from genotypes. I have been working on this for quite a while now, but one of the big challenges that I've always found is that whenever you want to do something around data with genotype to phenotype predictions, you have to beg people for data. You have to go to public databases, which are very heterogeneous, shall we say. I'll tell you a little bit more what I mean by the heterogeneous term here in a few minutes. Or at some point, you just basically lose patience and decide, well, you know what, I'm going to curate my own. And so that's what we did. And that's where this CABBAGE project came out of. So CABBAGE stands for something to do with comprehensive assessment and benchmarking of bacterial genotypes, or bacterial-based genotype. And so we were very lucky this year to finally secure some funding for it from the MRC, which is the Medical Research Council in the UK. And been able to hire 2 fantastic postdocs to work with me on this. And one of them is actually here. Loma, if you want to wave. Right, so any specific questions about the stuff that I'm going to be talking about, please talk to him, or just feel free to talk to him in the break. He's fantastic, and he's been really, really helping a lot with not just the CABBAGE work, but also this specific data set for the challenge. So let me just talk a little bit about the challenge this year. So this is our 3rd year, 2nd or 3rd, depending how you count. My 3rd year of being involved, 2nd year of sort of really being the curator, I guess, of the data for it. So 2023, the focus was data collected by the MetaSub consortium. Worked on that with Pavel, and we basically looked at, what genes were found in the different parts of the world. Can you say something about what part of the world that data comes from? So that was a really interesting challenge. A little bit different from the genotype to phenotype prediction. But it was a really interesting start, and we got a lot of interest from that. Last year, most of the data was for training was from Patrick, and for the testing data, we took it from publications. Now, the thing about publications is, surprisingly, quite a lot of them, quite a lot of the papers that publish data on genotypes and phenotypes for AMR do not actually make it into any databases. Okay, obviously they are in the PubMed and PMC databases usually, but they're not, the data itself, the phenotype data specifically, is not going to be in any of the databases that exist. And so we had to go through, and I'll show you a little bit about what we did in a minute, but we had to go through and extract some data from those papers, which of course is a lot harder to do than to do it from databases, but still possible. So, and then this year, we also threw in some training data from 2 other databases, NARMS and DRO, still trying to keep it relatively, homogeneous so that the training wouldn't be too, the training process wouldn't be too confused by the heterogeneity. But obviously, as soon as you have more than one database that you start from, even within Patrick, there's a lot of heterogeneity anyway. But once you have more than a couple of databases, it gets a little bit messier. Also increased the number of samples, went from 8K to about 11K in total, and went from 2 antibiotics or classes to 4. And then in 2026, which is next year, hopefully you'll all still want to participate. But at that point, we really hope to leverage the full breadth of the CABBAGE database. And that's what I'm going to talk about next. What is it looking like? We're making really good progress, given that we've only started in earnest in January. I'll show you why this is important as well. A few reasons. Of course, there's impetus to understand the links between the genotypes and the phenotypes. It's not always perfect, right? You cannot always get all the information that you need about the phenotype from the genotype, right? We know that in some species there's gene expression data that makes a difference, right? And we don't usually have gene expression data alongside the genotype. Sometimes we do, but very rarely, right? That's one. Another limitation is that when we have species that are maybe not as well annotated, again, or don't have good references available, also makes it harder. But also the other thing that we really care about at the end of the day, right? The reason we're doing this work is, well, two reasons really. Surveillance, which is kind of tracking how these AMR phenotypes spread around the world, but also clinical, right? Most of the interests, we're a very selfish species. We mostly care about ourselves. We care about how we can get better if we're getting infected with these resistant bacteria. So clinical prediction, figuring out which antibiotics should work, which antibiotics will not work. Now, of course, the problem is that even if you can perfectly predict the lab phenotype, which is to say, what is the minimum inhibitory concentration for a particular drug, or just, is it resistant or susceptible to this drug, it doesn't necessarily tell you the clinical outcome, whether, giving a drug that works is actually going to cure the patient, or sometimes you end up realizing, actually, we gave them a drug that didn't work or wasn't supposed to work, but still has somehow worked, right? And that's something that happens. I'm not going to give you any specific anecdotes about people, but last year my cat had a urinary tract infection, and it happens to cats as well. They got infected by stuff. She had to spend, it was a serious one. She had to spend about a week in a veterinary hospital. And they gave her some antibiotics and she got better. And then as we were leaving the hospital, the veterinary hospital, the susceptibility results came back. And it turned out that the bacteria that she had were resistant to the antibiotics that she got, but she somehow got better. So, it's obviously, okay, yes, in animals, it's even harder. In the veterinary space, the break points might be completely off. Fair enough. But the point is that it's still not a perfect predictor, right? So even if we got the phenotype perfectly, we could still not be as informative as we would like to be about the clinical side of it. So that's another gap that we have to bridge. But for now, let's just focus on the things that we can control. So the genotype to phenotype links, the published methods that are out there, unfortunately, I'm not going to name any specific ones, not going to single anything out, but they rarely generalize well, okay, beyond the training data. That is a fact. We've tried this in many different contexts and many different scenarios, and almost all of these methods generalize poorly. Okay, that's fine. I think that we just have to acknowledge that, right? We just have to acknowledge that in order to really get results that generalize, we should try to train on as broad and as comprehensive a data set as possible. That is exactly the aim in particular, one of the particular aims that we have been pursuing with trying to get all of the publicly available data out there. Google DeepMind, which has been in the news, of course, recently, well, especially Google Gemini, I guess, with the incredible performance in the International Math Olympiad, but such as it is, they are very interested in different applications of artificial intelligence. We talk a lot about artificial intelligence. I had a couple of meetings with them through the Fleming initiative at Imperial. They're very interested in getting involved. Okay. However, Pushmeet Kohli, who many of you probably know by name, sort of one of the leaders of that research side of it, said they would like to see something similar to a CASP challenge. Okay, the CASP challenge, as you all know, is the critical assessment of structure prediction. Right, this has been running since the mid-90s, has been very successful in kind of benchmarking methods for predicting the structure of proteins, and eventually, motivated them to try to throw their hat in the ring. Gave rise to AlphaFold, and now AlphaFold ++, whatever the latest version is, which is also publicly available, has been a huge, huge advance in the field, right? So, but the reason that they were feeling motivated to do this in the first place is because there was a an objective gold standard. And there was a community that was interested in kind of seeing the field evolve and develop further and get better and better, right? And so here, in a way, similarly, what we're trying to do here with the CAMDA challenge, right, is we're trying to create a platform, if you like, or a community, right, of people who want to develop methods, who care about the results, and who can essentially get us to the point where somebody who's maybe not as experienced with the field of AMR, either in the genotypic side or on the phenotypic side, can still take something that we've developed, training databases, standards, and so on, and can just get involved. All right. And then there is an extra challenge in comparison to protein structure, of course, right? We're not dealing with a static phenotype here, right? If we throw a drug or a drug class at a particular bacterial species, initially it might be susceptible. Over time, it will become resistant. And then when we stop, it will become susceptible again over time, right? So it's not a static phenotype. And it's not, again, another reason that this prediction is challenging is because, okay, it's not on the scale of years, it's probably on the scale of decades, but the phenotype does change over time. And also the standards for the phenotype, which is to say, what is the minimum concentration that you need to see as the MIC in order to call it resistant, that also changes over time. Right? So let me just very briefly summarize what was the situation when we started. So there were over 10 different databases that contained some AMR phenotype, genotype data, each with its own formats, idiosyncrasies, and so on. There was no unified representation of the phenotypes. The methodology used differed. The critical concentrations applied differed. In Europe, we have UCAST. In the US, we have CLSI. Elsewhere in the world, they tend to pick one or the other. Sometimes it's a third standard. Replicates are usually not reported. There's differences by source of isolate, whether it's blood or urine makes a difference. So what we try to do is create some order within that slight chaos. So we've extracted and processed data from all the major databases, including those 6, but also a few others that you probably know. And we also carried out a systematic search, literature search of papers, okay? Because that's the part that's missing. And actually over 20% of the current database contents are things that are found only in papers, okay? So they've never been, as far as we know, right? They're not part of any of the databases that we looked at. They're only found in papers. And so we had about 35,000 papers we looked at, and in the end, only about 150 of them were worthwhile in terms of the amount of manual effort that goes in. So I basically said anything that's below 100 isolates, I'm not even going to bother. Too much work, too little reward. Eventually, we're hoping to automate this. We're working towards this, but it's slow. And unfortunately, the majority of them only have R or S values, sometimes I for intermediate, not MIC values. Why is that a problem? Why am I sad about this? Because MIC values don't change their meaning over time, okay? Resistance and susceptibility do change their meaning over time. If you apply the same, if you're testing your isolate in 2020, chances are by 2025, the same, minimum inhibitory concentration could now classify it as resistant when it was previously susceptible, right? Or vice versa. So the breakpoints change over time, and that's a problem. Okay, so that's why if you're ever generating this kind of data, please report MIC values. Feel free to report the RIS values as well, but please report the MIC values. Okay, this is what our schematic literature search looked like. So we just had some terms for phenotype-related stuff, have some terms for genotype-related stuff, and had some terms for association-related stuff, essentially, right? So combinations of the two. And then we just had, multiple iterations of this, eventually got it down to about those 150 papers. As you can see, there's some blue. So blue means papers only. Red means both papers and databases, and green means databases only. And this is just by breakdown by isolate, okay, and by species. So number of isolates, number of samples, if you like, for each species. about 250,000 in total. And this is the entry. So what is an entry in our database is a unique combination of a biosample from the biosample database and an antibiotic, okay? All of this needs to be standardized, as Thomas, previous speaker, very correctly pointed out. Standardization of the data is hugely important. Unless you've standardized your data, you've got nothing that you can learn from successfully, right? Whether you're doing machine learning or whether you're doing, human-driven, trying to sort of understand what you've got there. There's a total of 58 species. We mostly focused on the bacterial priority pathogens on the WHO list. There's a few others that are sort of related or similar genus but different species, which we've allowed, but we're not necessarily going to prioritize them. About 160 antibiotics in total. The ones that you see here are the ones that have more than 1,000 entries. Not surprisingly, if you know what kind of antibiotics we use these days, right? So carbapenems are very prominent in this. It's a little bit small, so I apologize for the font. And in terms of the fields that we've added, so we've done a lot of manual processing of the papers to try to understand exactly how the phenotype was obtained, because that's important, right? Whether it's disk diffusion, whether you're doing broth microdilutions, what temperature you're doing this at, and so on, right? All of these details can matter somewhat to the outcome, so they're not always reported, which is why we have a lot of missing data. But this is a list of fields that we have. So we have some genotype fields, some phenotype fields, and some isolate metadata fields. And the bar represents the percentage completeness. Okay, so of course biosample IDs has to be complete, right? Because that's our kind of determining factor, the antibiotic name as well, and abbreviation. But other things we're allowed to be missing, right? And there's quite a lot of missing values, in particular on the isolate metadata, as you can see, host sex, host age, those kind of things, rarely reported, which is okay. We might not be able to analyze them, but that's okay. Right? We'll be able to analyze the things that we have more information on. So there's a little bit of a bias there, but by and large, it's relatively complete. And we're ready to release this. So we're currently in the process of integrating this with the biosamples database. This is version 1. I'll tell you a little bit about our next steps. So we want to automatically identify any additional relevant papers. Of course, we're not, deluding ourselves to think that this is all the papers that are relevant that we found. We've probably found a good chunk of them, but there's definitely more. Also, ones with fewer than 100 isolates, we still want to add them to the database. We want to process them. We want to try to do this in an automated way, in which case it will be worthwhile, right? If we're doing it by hand, as I did initially, then of course that will take too long. We're integrating it with the biosample database with ENA. We have a collaborator at the ENA on board, John Lees, who's been fantastic. And they're very enthusiastic about doing this. So very soon, I can't tell you exact dates, but very soon, probably by the end of this year, everybody will be able to look at a biosample that's a bacterial biosample, as long as somewhere the phenotype for it, an AMR phenotype for it has been reported. You'll be able to look it up. You'll be able to automatically extract it. You'll be able to look at all the isolates, all the samples for which there is information about this, and so on. We're trying to partially at least automate the curation process. It's slow. It's challenging, as you can imagine. We're collaborating with Phage. Some of you might know about it, the data structure group, to basically create an ontology that would describe the metadata better. At the moment, we're just trying to make the best interpretation of the data that we have. Also, eventually benchmarking the algorithm. That's some way of essentially extending the challenge, if you like, to a broader class of algorithms. So obviously, those of you who took part this year will have used your own algorithms, but we want to also try to benchmark a little bit more broadly. And then eventually, if we find something interesting, novel, we'll be able to validate that in the lab. One of my postdocs, Emily, has had a lot of experience with doing that. She's at the bottom right here. And yeah, I just wanted to acknowledge all the collaborators. This has been initially funded by FIND, the Foundation for Innovative New Diagnostics, before the MRC was in position to fund it. I also got some initial funding from the Sloan Foundation and Genome Canada a while ago for doing some of the background work for this. Fantastic collaborators, Anita and Swapna from FIND, Nicholas and Fernando, who did the initial benchmark DR platform, John and Rob Beardmore from Exeter University, who have been the main collaborators on this project, and then Hilma and Emily. And with that, thank you very much. Happy to take any questions, and good luck with next year's challenge.
Thank you, Danny. So maybe one, two questions, and more during the so yes, please.
Fantastic efforts. I really like your ideas about trying to have a cast-like competition, thinking about what that could look like. They should develop Micro, which I just presented a parallel sessions in knowledge graph for microbes where you source back ideas and antibiotic data. And thinking about the PDB, right? That's been very well funded in a kind of a self-contained format that has a sequence actually right inside of the structure. So do you have any thoughts about where we could go with our data, right? And maybe also fund some standardized experiments, right? And get more people involved in this effort.
Yeah, so that's a really good question. So I think that at the moment, the biosamples database sort of seems like a natural home because the phenotype data is not huge, right? It's in comparison to the genome, it's probably like 1% of that, space is occupied by the phenotype. So it's not going to be a big deal for them. I think it'll make it much easier to access. Eventually, of course, if you got a much richer phenotyping information, like if you're doing standardized experiments and so on, I think that we'll have to have a separate home. I'm not yet sure where that, maybe pathogen portal, maybe something else. So we'll sort of have to explore that as and when we get to that. In terms of, so in terms of actually assessing that in a standardized way, I mean, it'll be a lot easier to talk about that seriously when we can get UCAST and CLSI to agree about the breakpoints, right? Until that happens, I feel like it's a nice idea, but we're not quite there yet. But yeah, thank you very much. Looking forward to checking out the knowledge database as well.
Thank you. Okay.
Thank you for your presentation and work, and also glad to hear that the CAD is doing better. I was wondering actually in the same direction. My question is, would you see releasing also some sort of guideline or like requirements for people in the future to about how they should conduct the experiments and report the data, and especially metadata about all the technologies they use, so you can later add them to your challenge and increase more and more data and eventually get your foundation more.
Yeah, absolutely. Thank you very much. Very good question. And so the answer is absolutely. We are hoping that this. So I didn't quite go into the details of that, but the fields and the sort of underlying formatting for these fields is so it's already a refinement of what the NCBI offers and what the NA kind of offers. So it tries to kind of collect all of that together and add a few more additional fields. We would love for people who produce this type of data to submit it in that format. We're going to work with the World Health Organization. We have some connections there to try to get them to adopt this as a standard, right? Because I think that's really the only way to make it happen is if there's an authority like the World Health Organization that sort of stands behind this. We've had some initial talks with the lead AMR person there. They're interested. They're potentially open to the idea. So we're hoping to go there. We are also conscious of the fact that it's very hard to impose on the people producing the data additional requirements, right? Beyond making it public. So I always tell people, if you've got data in whatever format, just make it available, right? We'll deal with it. Of course, if you have extra time and extra resources, please annotate it carefully and please, fill out all these fields and please provide all this additional metadata if you can. But if that's going to stop you from sharing anything, then just share what you have. So, yeah.
Okay, so 2 quick questions, quick responses because we are
sorry.
schedule, so we can then continue the last.
Absolutely.
Okay.
real quick, plasmid phage in the cover database?
Great question. Not working on that at the moment. Very important topics. I can talk to you more about it during lunch. But yes, not working on it at the moment.
Thank you, also, Tom. I just want to ask if you're also collecting GROW, those response curves.
Those response curves would be great. I think there's, again, just like with the gene expression data, it's a lot rarer as a data type. If we've got those response curves available, we'll definitely plan to add them on. But that's not at the moment. That's not a focus. So yes, thank you.
Okay, thank you again.